Okay. Is it the full screen with the one slide?
Mhm.
So good morning, everyone. I know some people are still joining, but I wanted to welcome everyone to the 5th Boston Pharmaceutical Symposium. This is our second webinar in our series. Our next webinar is in 2 weeks, where Michael Hughes from the Center for Biostatistics and AIDS Research will be talking about design considerations in a COVID-19 platform trial. And then we'll have 2 more webinars in October. I also want to bring your attention to our website. So this is hosted by the ASA Boston chapter. We have a bunch of other events, including a short course on adaptive design next Friday. So please visit our website and look at other upcoming events that are going on within the Boston area. Most of the events right now are virtual. So next I would like to introduce our president of the Boston chapter of the ASA, Olga, who will be giving some background on how to use the Zoom.
Thanks so much, Kristen, and thank you everyone for joining us today. My name is Olga Vitek. I'm one of the co-organizers and also president of the Boston chapter of the ASA. I'm very excited to see so many of you joining us this morning for the webinar. And as Kristen is saying, please take a look at the website because we have additional events which are not listed on the slide right now, and there are many other interesting things which are organized by the chapter. But for today, I'm really excited to host our webinar, and I am glad that we can do it over Zoom. It would be nice to be it in person, but Zoom has some advantages, so let's take advantage of that. So I'm sure all of you are familiar with Zoom, but you may or may not be familiar with the webinar part. So if you look at the bottom of your screen, there is a chat box. We will use this chat box only for things such as I cannot hear or some just logistics questions. But for the science part, I hope that you will be using the Q&A box, which is also at the bottom of your screen. So we hope that the webinar, even though it's on Zoom, it will be as interactive as possible. You will have as many questions as possible directly related to what is presented, but maybe also somewhat related. So please use Q&A for that, and our two speakers, Rachel and Jen Chang, will be able to answer that. And we also have a possibility to promote some of the attendees to presenters so that you are able to speak and share screen as needed. So as we go on, if this becomes necessary, this is also possible. So we hope that even though it is on Zoom, it will be as interactive and as useful to you as it can be in this format. So that's all I have to say. So now let me introduce Jen Chang, who is one of the chapter officers who will talk about the plan for today and will introduce the main speaker. Thank you so much.
Thank you, Olga. Thank you, Christine. Good morning, everyone. Welcome to today's webinar on adaptive design for optimum dose determination in IO and the cell therapy, which is also part of 5th Boston Pharmaceutical Symposium. My name is Jen Chen Lin from Takeda. It's my pleasure to introduce today's speaker, Rachel Liu, who is also my colleague from Takeda. Rachel is an associate director in the statistical and the quantitative science department in Takeda Pharmaceutical Company. Her research interests include dose finding design, adaptive design, mass protocol, machine learning, and real world evidence. And he has also served as statistical leader for multiple early and later phase oncology and cell therapy program. And she's particularly passionate about bringing innovation to drug development. Just Olga mentioned, before we start presentation, a few additional housekeeping items. Please feel free to mute yourself during the presentation and enter your question or comment in the Q&A box in the Zoom link, and we will answer those questions during the middle of presentation or Q&A at the end. So without further ado, Rachel, please take over.
Thank you. Thank you, Jen Chang, for the nice introduction. Thank you, Olga and Christine, for organizing this webinar. It's really my pleasure for this 1 hour session that we can talk about our thinkings, recommendations, and our recent works on this very important topic, which is optimal dose determination in IO and cell therapy. And during this 1 hour session, I'm hoping we can cover from different aspects. We'll be starting talking about the designs that focusing on the safety, either in the cycle 1 or late onset toxicities. Following that, we are hoping to introduce some more advanced scenario work and our recent works when we're talking about the dose finding in a basket umbrella trial setting, and when we talk about dose finding incorporating multiple endpoints. So as Jen Chang mentioned, please enter your question through the Q&A, and I'm hoping we can cover all those your questions during the presentation. So as everyone knows, the IO and the cell therapy has been a booming area for development globally, and actually the recent approvals from IO further stimulate this area. And from this review, you can tell that globally, many pharmaceutical companies and institutions have shifted their focus in the oncology development into the IO. And also the cell therapy as one very important largest growth category within the IO has been rapidly growing. The recent cell therapy approval, including on the lymphoma and the multiple myeloma, but actually if you look at the current competitive landscape, you could find many companies are pursuing the cell therapy options in oncology beyond the CAR T cell and including TCR, natural killer, and etc. Due to the very special scientific rationale for both the IO and the cell therapy, there's of course many emerging questions arise in this area that conventional oncology development are not able to address. Recently, FDA published a guidance to encourage sponsors to have more thorough thinking and comprehensive looking in terms of the early phase clinical trial design of the cell therapy, which we can cover some of the major considerations in the later slides. So let's talk about the dose finding. As everyone knows, oncology in general, the dose finding is already complex compared to other therapeutic areas. We do dose finding in patients, not healthy volunteers, and conventionally the dose finding focuses on to identify the dose limiting toxicities in order to find the maximum tolerated dose. At the end of the dose finding, the conclusion will be based on the MTD recommendation. We can provide a recommended phase 2 dose, which is going to be incorporating other sources of the data, for example, efficacy, PK, and PD. And there are many different types of designs has been developed in this area. But is it good enough that we can just leverage whatever have been developed in oncology and applied into the IO and the cell therapy? Unfortunately, the answer is definitely is not good enough. In the IO and the cell therapy, we should think in a more patient-focused way for the dose optimization. And we definitely should ask ourselves, before you enter into the clinic, what are the major objectives you want to obtain from the first in-human trial from the IO and the cell therapy? Do we think the DLT in cycle 1 is still appropriate for the objective of the IO and the cell therapy development? Do we think we need to consider more than the safety in terms of the dose finding, meaning we are going to having a more good start for the future development to improve the probability of success? Do we consider MTD is still the primary endpoint we should be pursuing in the dose finding? And also, most importantly, during the many companies has been pursuing in accelerated development fashion. So with that said, there are many hidden elements we have to consider during the design thinking. For example, is the patient population heterogeneous enough, and we need to think about how to select the patient in the next phase of the development during the dose finding. Can we consider in a more seamless fashion to combine the phase 1 and the phase 2 so we can further reduce the timeline and bring fast innovative therapy to patients? And also, is it the current dosing schedule good enough? Do we want to consider other dosing schedule? Do we want to consider other combination partner selection into the dose recommendation? So there are many pending questions, and in IO and the cell therapy, which is more prominent compared to the conventional dose finding, and which meaning the phase 1 trial, what the phase 1/2 trial plays a more critical role in terms of the future development. So in these slides, I'm trying to list several special challenges in the IO and the cell therapy compared to the other pipelines in the oncology in general. Of course, I'm sure when you work on IO and the cell therapy, you must think about a more comprehensive list than what we are putting here. In general, the risk of toxicity in IO and the cell therapy is more concerning compared to other therapeutic areas. It's not only talking about the DLT in the cycle 1, because of the prolonged biological activities in the IO drug, it is not uncommon that the patient is going to having the immune-related adverse events that's going to happen later in the cycles. And also the animal models is often not very informative because the way the IO and the cell therapy drug is working, it utilizes the human immune system. So it's of course, those animal models will not be able to generate a good prediction, no matter regarding the dose ranges or regarding what's the possible most potential therapeutic dose level going to be. And the IO and the cell therapy, in generally speaking, the exposure response relationship, specifically the dose and the toxicity, the dose and efficacy, such relationship is much more complex. It's going to be a naive assumption to assume both toxicity and efficacy don't going to increase with the dose level. So with that said, the conventional dose finding framework might not work in this area. And in the cell therapy specifically, things could be even more complicated. We all understand that the manufacturing in the cell therapy is a critical part, and it's actually very complicated. It often lead to very limited dose level for you to explore during the initial of the phase of the trials. Different versions of the products naturally can generate many variabilities that you need to consider in the trial. And the patient population is often all comers, so there's also heterogeneity in the patient population, etc. The estimate need to be considered because not every patient is going to be receiving the cell therapy during the waiting time, etc. So there are many problems there, and that's why we think it's important that we very much leverage what we have developed in the oncology, those findings, but we need to think in a more deeper way so we can laser focus on the questions we want to address here. So long word short, the conventional thinking to identify DLT to find MTD may not be optimal for the cell therapy and IO development. And not only pharmaceutical companies trying to think about a more smarter way to proceed in IO therapy. In the recent time, the FDA officials have been quite vocal to advocate a more comprehensive dose searching. It raised some concerns that the oncology sometimes are rushed into the dose finding phase, which meaning you may have to revisit toward the finish line because different dosing schedules or different dose levels might also work, etc. So with that said, in the following sections, I'm hoping to cover the different considerations we just talked about to tackle the challenge listed earlier. Okay, so let's talk about the dose finding designs that can still be using the DLT information. So as I just mentioned, not every IO and cell therapy, the DLT in cycle 1 is appropriate, but when you have a pipeline and you believe the DLT in cycle 1 is appropriate, there are actually different kinds of designs that you can consider during the dose recommendation and the dose searching path. Obviously, 3+3 is no longer optimal. It has been proved having the low probability and high variability in terms of FTD estimates. It is also not very operational convenient because it's sort of tied to the 3 patients cohort, and we all understand during the practice that could make the patient enrollment delay. For example, some patients might be dropped due to operational reason, and you have to enroll more, or you cannot just enroll more than 3 at a time. And with that said, improved designs has been developed in the previous decades. In general, it can be considered into the 2 major categories. The first one would be the model-based design. One famous example out of the model-based design is the Bayesian logistic regression model, shortened as BLRM. And the model-based design, we are going to assume a specific dose DLT relationship. So with that said, based on this assumption, the model can provide the prediction what estimates of the DLT rate at each dose level. Another category of the dose finding design is the model-assisted design. In the model-assisted design, you don't have to use all those levels of data, which meaning you don't have to assume a specific relationship between the toxicity and those levels. And some famous design under this model-assisted design category includes MTPI, MTPI 2.1, etc. And one very good feature of the model-assisted design is it can provide the transparent dose escalation decision table before the trial, which often the case we feel very good feedback when we apply that and from the investigator and the clinicians. So here is the BRM design in the model-based design category. And this is the formula or the dose DLT relationship, the BRM model applied here. And specifically speaking, the BRM is a Bayesian framework. So you can actually having the previous study data or even the animal data to be incorporated as a prior information. And then with observed data and this specific assumption of the relationship, the BRM model going to be generating the posterior probability of the true DLT rate falls into 3 intervals, which is overdosing interval, targeted toxicity interval, and underdosing interval. And with that said, the dose recommendation will be provided that having the maximum posterior probability that the true DLT rate falls into the target interval. And this interval, scientifically speaking, should be the interval that is tolerable and also can provide the most potential therapeutic effects. On the other hand, the BRM model can also apply the overdosing control criteria, which meaning we are going to eliminate the dose that going to having a higher probability that the true DLT going to fall into this overdosing interval. So for studies using BRM, you use all those levels data. And the advantage here is not only you can provide the estimates for the dose level you have explored, by leveraging the specific dose DLT relationship, you could also predict the posterior probability of those 3 intervals for the dose levels you haven't been explored. And also, you can also, if you want to try some ad hoc intermediate dose levels, and the BRM model could also be a tool to help you select the most potential or the most appropriate intermediate dose level you want to do here. So obviously, those flexibilities adding together going to having a good feature and a good functionality for the dose for your dose recommendation and the final MTD estimation. But of course, this specific dose DLT relationship need to be carefully examined. If it's going to be violate this relationship, the dose recommendation from this model will be misleading. And then I also want to give a very high level overview about several model-assisted design. So MTPI is one type of the model-assisted design proposed by Dr. Ji. And specifically speaking, the model-assisted design does not need to rely on the dose DLT relationship, so it's only looking at the current dose data, and it still defines an interval in terms that the target DLT going to be generated from this interval, and that interval is considered for the MTD recommendation interval. And the way MTPI selected the dose level is it will calculate UPM. And based on the UPM, it is basically the posterior probability of that region divided by the interval. And in this way, the dose recommendation will be provided considering which interval having the highest UPM. And in the final dose recommendation for the MTD, it will be selected retrospectively to consider the estimated DLT rate with the target DLT rate. The right side is an example for the dose escalation decision table that can be generated before the trial starts. Basically, you need to define what your targeted MTD rate should be. You need to define what's the maximum sample size, and this table going to generate it by different scenarios of number of patients treated at each dose level and the corresponding toxicities. And by reading this table, you will know in certain scenario what's the dose escalation decision would look like. But by looking into this table, you may find out some of the results might be controversial. For example, if we have 4 patients dosed at this dose level, and we observed 2 DLT out of the 4 patients, which basically 50% of the observed DLT rate, the MTPI model is still going to provide a stay recommendation, which of course, if we do not have strong belief of this dose level toxicity profile, it could generate aggressive decision making. And the reason for providing such recommendation is due to the consequence of Occam's razor. And to address that, Dr. Ji's team further improved MTPI design into the MTPI tool. Basically, is to make sure the intervals is further break down into an equal length. And in that way, the dose recommendation decision table, for example, in the right side, will be able to avoid to provide aggressive decision making. And now, for example, the 2 DLT out of 4 patients will be a de-escalation instead of a stay. So I think MTPI series design definitely is a good option that when you are not sure about the dose-DLT relationship and you want to generate a very transparent decision table and to be used to communicate with the team. And MTPI2 definitely has a good feature in terms of this purpose. Another category, another very popular design under this category is the Bayesian optimal interval design. So the Bayesian design has a similar philosophy as the MTPI series, but it definitely a different way to generate the dose escalation and de-escalation decision tables. The way it is working here is in the Bayesian design, we are going to having 3 hypotheses. The first one is the true DLT rate for this dose level is the targeted DLT rate. And the second one will be the true DLT rate in this dose level is in the underdosing DLT region. And the last one will be the opposite direction, which meaning the current dose is in the overdosing region. And the way Bayesian work here is it will generate boundaries along the E and along the D. And once the boundaries are generated, you just need to compare your observed DLT rate with these boundaries. So you can provide the dose escalation or de-escalation decision making. And the boundaries is generated by utilizing the Bayesian framework, by utilizing this hypothesis to calculate the probability of the decision errors here. And the boundary will be obtained by minimizing these decision errors. So this table is an example that having a Bayesian design with a targeted toxicity rate of 30%, the boundaries will be generated as 20.236 and 0.358. And converting to a more granular way, and this table showing that under different number of patients treated at the current dose, what would be the de-escalation boundary looks like in terms of the DLT numbers. So technically speaking, the Bayesian design is going to be provide very similar dose escalation decision compared with MTPI2 design. So either one would be a good choice that if we are talking about cycle 1 DLT dose recommendation. And some more advanced version still in the cycle 1 DLT framework has also been developed. For example, Bayesian design is a good one that you want to incorporate your previous study information, which meaning, for example, if you are having a single agent trial ongoing and you're already having the data for a good range of the dose levels, and then you plan to having another dose finding trial, single agent, but with different dosing schedule, and you don't believe that new trial will, that different dosing schedule will generate substantial more toxicity compared to the previous trial. So you can use the Bayesian design. And then the decision making will be dose specific, which meaning for the doses that has been explored in the prior study that has been deemed safe, the dose recommendation for that specific dose for the future trial will likely to generate a more aggressive decision making compared to the Bayesian design. Vice versa, if a specific dose in the previous trial has been considered having alerting toxicity profile, then your dose recommendation for the future trial using the Bayesian going to generate a conservative decision making compared to the Bayesian design. So this Bayesian I do recommend for such sequential development in a specific agent in the pipeline, because it can best use the information what you can have, and it can definitely providing more efficient decision making in that sense. And other designs which we don't have to cover in detail today, but I just want to read out the name, and you can read through their papers if you are interested. Having a keyboard design, I3+3 design, or cumulative cohort design. So as a summary, the DLT in cycle 1, not every IO and cell therapy appropriate to use DLT in cycle 1, but if it is appropriate, there are multiple design options has been developed here. And I just take some favorite ones and good features ones to review in this section. But of course, to decide which one to be used, we need to have statisticians need to have a good judgment beyond the statistic itself. You need to have a good understanding about the potential toxicity profile or some hypotheses for this specific candidate drug. And also you need to understand what is the planned dose ranges. If you have a long planned dose ranges, and you think the monotone relationship is still likely to be reliable, then BRM definitely is a good choice. If you don't have a long dose ranges, or if you think the assumption between the dose and the DLT from the BRM model may not work here, then those model assisted design like BOIN MTPI2 will be a good options here. And of course, each design should be examined through simulation practice and close collaborate with their team to generate some scenarios that are going to cover the potential cases of this drug. So moving forward, let's talk about something more than the DLT in cycle 1, and which meaning that late onset toxicity is something you may want to consider in the IO and the cell therapy development. So actually, the IO trials, many IO trials actually have the risk of the late onset toxicity. And with that said, if you just doing the dose finding trials using the conventional way of focusing on DLT in cycle 1 only, and in the right side of example, you may find out that when you find out those late onset toxicities for the previous dose level, it may already be too late to protect your patients dosing from the higher dose level. So with that said, late onset toxicity is something we definitely cannot just be naively handling as the other conventional dose finding trials, but you still need to have a very good specification or definition beforehand. For example, we need to work with the clinicians to understand for this late onset toxicity, what is the window we are talking about? It's not going to be cycle 1, but is 2 cycles enough or 3 cycles enough? That's going to decide how the dose recommendation going to be looking like, considering the maturity of your patient's DLT data. And also, we still need to consider when we consider the late onset toxicity, you still need to define what your target toxicity rate looks like. Is it 30% good enough, or is it a little conservative that we could increase that a little bit? So with all those definitions and the discussions ongoing before you are trying to provide a good design, and then that's going to helping you to consider if the late onset toxicity design is going to be feasible for your trial, and also what's the operational implication looking like. Of course, for the late onset toxicity, no one going to prefer we just wait for example, 3 cycles until we are having a dose recommendation decision. We want to have as much as possible a seamless decision making, and as least as possible the enrollment path. So with that said, again, in the late onset toxicity design recommendations, there are 2 major categories. For the model-based one, tight CRM is a very famous design in this setting. You still need to assume a dose DLT relationship, and during the estimation, it will assign the weights to the outcome based on the maturity of the outcomes. And another category is the tight BOIN design. It is a modified extended version on top of the BOIN, which meaning there's no dose DLT relationship is needed. And on the other hand, they will define STFT, a standardized follow-up criteria, to evaluate for the current cohort if you have enough maturity of the information for you to make a dose recommendation decision. So both designs are going to be a good option when you consider the late onset toxicity in terms of the dose recommendation. So Jianchang, if there's no emerging questions you want me to address here, we can move on to in a more advanced scenarios for the dose recommendation in IO and cell therapy.
Just maybe one question related to the dose optimization initiative you mentioned before, because it seems there's an increasing interest in this field. So a question is, do you envision it will become more and more common, in future when we design dose finding design, it will be more common we explore multiple dose cohort in phase 2 trials?
Yeah, definitely. I think we are definitely more and more open to the idea that multiple dose cohorts need to be considered in terms of the RP2D decision making, and also different dosing schedules will also need to be considered. And not mentioning, if you are talking about combination strategy, different partners you want to consider. In IO and cell therapy, the preclinical model or the data is not very informative. So the weight has been heavily shifted into the patient's trials, and to understand if certain dosing schedule going to work well, or if certain dose is really going to generate enough confidence for you to move forward. So with that said, the conventional way of doing is more like a two-step or sequential. So you start from one dose schedule, one combination partner, for example, and one group of the population, and then at the end of such dose finding trials, and then the second step will be, for example, open expansion cohorts to explore different possibilities. And in the next 2 sessions, I actually want to challenge this conventional thinking is, is it really appropriate, or is it really efficient enough to consider in such sequential order for the development? Can we thinking from the beginning to have a list of what we are trying to address here and trying to do things in a more systematic way? So we can cover those in the next 2 sessions about some examples.
Thank you, Rachel. I think the answer is very helpful. We actually have 2 more questions. The first question is related to the late onset DLT. So question is about how do we determine the appropriate length of observation window? So usually how long is sufficient for the late onset DLT? Is it typically 2 cycles or 3 cycles?
Right, but I think that's a very good question. I think that's also something as statisticians we should continue to discuss with their clinical teams, and to basically emphasize the late onset toxicity design have the flexibility to consider the DLT beyond cycle 1, but the flexibility has a limit. You still need to clearly define what type of the late onset toxicity you are talking about that is going to be of concern for this specific drug development. And with that said, what is the possible window we are talking about? At the end of the day, it's going to be a decision to be made, and might be a compromise considering limited data, considering your operational timeline, etc. But a decision has to be made to clearly define why you want to consider late onset toxicity and what type of the AE is considered to be late onset toxicity. And scientifically speaking, how many months is, or how many cycles that we are likely to observe those late onset toxicity. That could be generated from historical data, from scientific theoretical hypotheses, or even from your preclinical model exploration.
Okay, one more question before you move forward, Rachel. So the question is about to find the optimum dose, how many dose level we need to test, and how many patients you will recommend at each dose level.
So that's also a very good question. The dose levels, as I just mentioned, that that's why we need to have a good understanding, not only from the clinically speaking, and in our experiences, the dose levels is definitely generally proposed by clinicians, by our preclinical team. They may have some data collected from animal, and they may have some preclinical models run, like those DMPK models, to project what is the possible dose levels we want to explore in the human. And with that said, if by understanding those rationale of the proposed dose level ranges, you can decide what models is going to be more appropriate. If you're only having 3 or 4 dose levels, and the BRM model might not work well because you may not have enough data to establish the approximation of this dose-DLT relationship. And then maybe the model-assisted design going to be a better choice in that sense. And also in terms of the number of patients at each dose level, in practical, people like to do things in approximately size of 3, which meaning, you may enroll 4, you may enroll 2, but there's definitely no good answer on that. I think the statisticians should be a safeguard in terms of not just thinking about timeline to putting very small number of the patients in certain dose level to do things in an aggressive way, and how to defend our position. That's why I was recommending and emphasizing the simulation practice is going to be very important. You are going to discuss with a team to generate different scenarios about the dose-DLT relationship, and through simulations, you can play with different number of the patients in each dose level, and you can see how the results going to be looks like, which meaning we are going to comparing the reliability. What's the chance you accurately estimate MTD, and how many patients you are putting there that is on the toxic dose level, etc. And through such practice, with the limited information we often have at hand when we enter through the clinical, it's going to help you to make a more rational decision making for the design.
Great. There's a few other questions, but I think we can wait until the Q&A section because I think we have 2 more sections to cover. Thank you, Rachel.
Thank you, Jianchang, and thank you for all the questions. So moving forward, let's talk about more advanced scenarios for the IO and the cell therapy dose finding. And as you know that in oncology, we like to do things in a more exploratory way in the beginning, which meaning in the first in human trials, according to the IE criteria, we are going to enroll. We're often going to enroll a basket of the indications, especially in IO. We are going to enroll a list of tumor types in the dose finding. And with that said, the heterogeneity in patient population might just be ignored. Different tumor types may not really necessarily having the same dose level as the most appropriate dose level to be considered moving forward. And also when we're talking about different dosing schedules, different combination partner explorations, we also need to think about if we really want to do things in a sequential way, or if we can actually moving those a little ahead, or actually ahead to your first in human trial for the single agent, and to consider how we can leverage the information each other. So this seems like, so that's why we are thinking maybe we should consider a master protocol concept instead of just doing things in all-comers conventional way. And this actually going to be a very important question to think about to avoid at the end of the development of specific indication that you have to revisit some dose schedules or some dose levels. For example, there's an example from Amgen, and FDA actually requested Amgen for this drug that to explore a lower dose level than the one that is approved in the label. So with that said, we need to carefully plan in the beginning. We probably already know what kind of the options in terms of dosing schedule, combination partner, or patient population we have to explore anyway. And can we having a more holistic thinking, systematic thinking in the beginning, so we can provide a more reliable recommendation moving forward. And with that said, I want to talk about using these unconventional thinking, specifically in the master protocol concept. Maybe we can having a basket trial, umbrella trials to be implemented early on. Of course, as mentioned earlier, no matter you do basket trial or umbrella trial, the commonly used in practice will be either naive pooling, which meaning you still ignore the patient heterogeneity or the strata heterogeneity when you do the estimation, or if you just do that in the fully stratified way, which meaning you are not really being impacted by other stratas data, but on the other hand, the limited sample size for each sub-trial or each sub-arm going to generate high variability, so going to be making your recommendation very, very challenging. And with that said, in the basket trial setting, for example, we can make those findings and enroll different indication as a separate arm in the basket trial concept, or if we are talking about different dosing schedule or combination partner, we could also do in umbrella trials. And that's just the design. It's not talking about the estimation. And in estimation wise, we are proposing to using a borrowing scheme, so we can leverage information across the similar arms, and that will definitely increase the efficiency, reduce the number of patients expected, accelerated the timeline, and it could also help you to get a more accurate estimation in terms of your dose toxicity relationship, as long as you are borrowing appropriately. So there are some design options in this category. Exchangeability, non-exchangeability model is a good option there. The EX-NEX design have a broader use, not only in the dose finding, but it can apply well in the dose finding. And in the EX-NEX design, the way it is work is it's going to be basically putting patients into two clusters. Either the first cluster meaning homogeneous, which meaning the strata in this cluster going to share the same distribution. And of course, the other cluster will be more heterogeneous, which meaning they're going to share different distribution. And how to assign the patients, and it's going to be dependent on weight P. The weight P is pre-specified. You cannot really estimate it from the data. So with that said, the EX-NEX model on one hand can borrowing in a more smart way to avoid over borrow, but it also highly depends on how you assign the weight P. And to overcome this hurdle, we actually propose a different way when we talk about those findings in basket trial and umbrella trial early on. And when we talk about we want to borrow the information in appropriate way. So we propose to use a Bayesian semi-parametric design. We name it as the BSD, and for the dose finding in such framework. And specifically speaking, we use the non-parametric Dirichlet process for the prior approximation. And for people who may not be fully familiar with Dirichlet process, it's actually a very popular technique in the machine learning, has been heavily used in the computer vision. It does not really assume a specific distribution. It's generated from a group of the distribution, and it's going to be adaptively learned from the data. So the left side is just an example to showing that using the Dirichlet process, it is capable to fit a broad spectrum of the distributions. So any shape of the distributions due to the machine learning, the learning feature of the Dirichlet process it can fit very well. And using this Dirichlet process into the dose finding, we actually break down our BSD design into two variants. The first variant, we call it as the BSD1. We propose to use this variant when you consider across different strata, you still having a belief of relatively homogeneity. So with that said, to skip all those technical details due to the time constraint, basically through the Dirichlet process, the distribution, the prior parameter distribution from each strata are going to be drawn from a relatively similar clusters. So here, and also when we talk about the clusters, these actually are implemented by the Chinese restaurant process. So with that said, through this dynamics, let's say we have 5 strata, and for the strata that through the CRP implementation, that's going to be assigned in the same table, and they are going to having the same prior parameter. And for the strata sharing different table, they are going to having a different value, but following a same same group of the distribution. So with that said, the BSD1 can interpret certain level of the heterogeneity, and also if you consider the homogeneity is also something going to be happen, and it is going to be a good choice. Another version of this BSD model, we call it as BSD2, which is going to be a more flexible use when you consider your strata are going to be rather heterogeneous, or if you do not have any prior information in terms of the heterogeneity level, then we can use the BSD2 design. And specifically speaking, the BSD2 basically is adding more layers in terms of the hyperparameters, and still using the differentiated process and the Chinese restaurant process, and which meaning for the clusters that assign, for the stratas that are assigned in the same cluster, they are going to follow from one distribution but different value. For the stratas that sharing different clusters, then they are going to follow from different distributions. So in this way, of course, it can account for more heterogeneity from the trials. And here is an example that using the BSD1, BSD2, and the EX in the dose escalation decision making. So let's assume we have 3 scenarios, and we have 3 indications in each scenarios, and we have the DLT data observed and listed here, which you can tell the heterogeneity level is going to be increased across the scenarios. And with that said, by using the BSD, BSD2, BSD and EX model to approximate the distribution, and you can tell that using the BSD1 and the BSD2, when the heterogeneity increase, it can account for more diverse distribution compared with the EX model. That's going to provide a more reliable estimate. And here is an example to further illustrate how the decision making going to look like compared with different types of the designs. Due to the time constraint, let's just, for example, focusing on the second scenario. Let's say we have 2 dose levels for the 3 indications, and this is the DLT data we observed from the 3 indications. And you can tell here that at the second dose level, the indication 1 having 1 DLT, while the rest of the 2 indications do not have DLT like at all. And using different type of the models, BSD1 and the BSD2 are able to capture the heterogeneity here, and thus to provide more indication-specific dose escalation decision. So our model going to recommend to stay in the indication 1, and still going to recommend to escalate for indication 2 and 3, while the other models is more like overborrowed, that all the decision makings are the same across the different indications. So with that said, when you talk about IO and cell therapy development, it is very common that you want to explore different possible tumor types and what different treatment candidates. And in the cell therapy, we may talk about different versions of the products. And so with that said, appropriate designs need to be considered. It's better to consider now than in the future that you have to revisit and create a lot of painful time that you have to revisit the label and having post-commitment trial, etc. And with that said, we are recommending to using a master protocol concept from the beginning. For example, using basket trial, umbrella trial through the borrowing dynamics. BSD is the one we propose, and EX model, which is also good options if you can clearly specify or comfortably specify the distributions carefully specify the borrowing amount by assigning the weight there. And if you want to have a more flexible use, our proposed model definitely can avoid misspecification and it using the machine learning technique, so it can adaptively learn from the data. And the last section, I know we are sort of have 10 minutes left. And the last section I want to cover is actually an emerging topic that can also address FDA's earlier advocates in terms that we may want to consider a different dose fluoridation, not only on the safety, but also on the activity wise. So with that said, the reason in IO and the cell therapy, and especially in the cell therapy, we may want to consider the dose finding more than DLT is because the MTD is not often the optimal dose. And based on the published data from the current available treatment options in these two areas, you may find that the dose activity relationship may not be monotone anymore. It could be reached plateau over a certain dose, or even some lower dose could generate similar efficacy or activities compared to the higher dose. So with that said, the conventional thinking for the MTD exploration is built upon the assumption that both the toxicity and efficacy will be increasing with the dose level increase. So with that said, you don't need to worry about anything. You just need to focus on the safety as long as you can provide a maximum tolerated dose, and that dose is going to be a good candidate for the therapeutic, most potential therapeutic dose level. And that assumption clearly has been violated in many IO and cell therapy drugs. And with that said, we should think about from a different concept that we are no longer pursuing to achieve the MTD. We need to think about a different way, and we call it as optimal biological dose, OBD. But of course, it could have different version of the name, but in reality, what we are talking about is a dose level that is tolerable, and on the other hand, it can generate the highest possible activity among the tolerable dose levels. And with that said, there's several design options that we can consider to incorporate the safety and efficacy endpoints from the dose search, which is actually important, right? Because if you do into a two-step way, which meaning you only do the dose searching based on the safety, and then based on the tolerable dose levels, you are trying to add in more patients in several selected tolerable dose levels to consider what should be the OBD looks like. That is very inefficient. That is also basically make your timeline longer than necessary, and also it can risk the patients, because in that way you may put the patients in some not very potential working dose, and that's going to be not reflect a patient-centric principle. So with that said, there are several design options. Again, following model-based design, there are FTOX design, SHH design, etc. Need to assume a specific dose, DLT, dose efficacy relationship, and even the correlation between these two endpoints. So it's hard to justify. And the model-assisted design only talking about the current dose levels. We have TEPI, 0.80, 0.12, and also we propose the TEPI2 and the UBI designs. In general, it's carried over the good feature of the model-assisted design that you can have a transparent dose escalation decision table to be generated before the trial. It also requires some sort of trade-off utility functions, so you can balance different combinations of the safety and efficacy outcomes. So just to quickly touch base the TEPI2 and the UBI design. The TEPI2 design is we consider that's an improved version compared with the TEPI design, which is trying to address some unrealistic decision making due to the Occam's result by breaking down the intervals into a more equal space. And then this input table is we can solicit from the clinical team, and that's going to be a guidance for us to consider what's the trade-off, what's the what kind of the combination of the safety and efficacy is favorable and what is not. And then the decision table is going to be generated following those inputs such as target toxicity rate, minimal acceptable efficacy rate, etc. And just as a comparison, the TPI2 table in some cases definitely is going to be providing more comfortable decision makings. For example, you don't have to escalate when you are having very good efficacy already, because we don't assume the efficacy definitely will increase with the dose level. And at the end of the dose escalation, the TPI2 will be retrospectively select optimal dose by using the histonic transformation and by utilizing the utility functions here. And the optimal dose will be provided with the one having the maximum expected utility function. And the UBI design is another design we proposed for the dose finding, incorporating both the safety and efficacy endpoints. It is extension to the Bayesian design, which consider the safety only. And the way it is working is, again, we are going to construct some utility functions, and then we can balance the safety and efficacy endpoints, and we can generate the decision table as is showing here. And in general, the UBI design and the TPI2 design are going to having a very similar feature in terms of the dose escalation decision making. And optimal dose is also going to be selected retrospectively here. And we put some simulation results here in the slides, which basically showing when we compare with different types of designs, how the results looks like. The F-talk sometimes works well, but for the cases that you are violating the pre-specified assumption, again, it's very hard to justify in the beginning of the trial. The F-talk is going to providing some not very good results here. But our TPI2 and the UBI in general has the best performance or comparable performance with others. And most importantly, our designs can ensure that we are going to allocate it very small number of the patients above OBD. So here is a hypothetical example how the design is going to work, which I don't think we have time to cover. So basically what I'm trying to say here is using the UBI, TPI2, or even other types of design, we actually can break the barrier between the phase 1 and the phase 2, or even break the barrier between the dose escalation and the extensive expansion cohorts, and trying to think the drug's profile in a more holistic way in the beginning, that of course going to reduce the number of patients that is needed. So I guess as a recap, I think in IO and cell therapy, the early development is playing a very critical role to determine the probability of the success. And also IO and cell therapy, the scientific questions is more prominent, considering the preclinical models are not very informative, considering the mechanism is often very complex, the interaction of the immune system, considering those assumptions, we keep using that the monotone relationship between dose safety, dose efficacy is no longer there. And also considering it's quite often we want to think about different dosing schedule, different patient population, different combination partner. Therefore, in this 1 hour session, just quickly wrap up, we are trying to think the design in these areas into different aspects and hoping to cover, as more comprehensive as possible. Is we are talking about how to handle the design in DLT and beyond. We are talking about when MPD is no longer appropriate, we want to use the OBD and how to do that when the design considering multiple endpoints. We also talk about to utilize the ballroom scheme and apply it into the basket trial and umbrella trial early on, so we can have a more patient laser focused decision recommendation. So again, well begun is half done, right? So we need to think very carefully, and the early phase design in the IO and cell therapy is not something we just do because we have been doing that in the past decades. We need to think carefully and smartly, and we have many examples that are showing if you do not think thoroughly in the beginning, you may have to revisit in the end anyway. So I'll pause here, and thank you for everyone's attention and the questions.
All right, thank you, Rachel. Great presentation. We did receive quite a few questions, although I understand we are kind of running out of time, but maybe we can stay a few more minutes to address those questions. So first question from Lam Zhou. So the design you mentioned before for IO are they also recommended for the cell therapy development? I think this is referred to late onset toxicity design part.
Right. So I think the cell therapy in oncology is a big pillar under the IO development. So with that said, many considerations in the IO will be or can be carried over for the cell therapy, like late onset toxicity. That's why I'm saying to understand that the potential profile is very important. In IO, we could have late onset toxicity. In cell therapy, we could also have. So with that said, it really depends on what is the scientific concern embedded there. And that's why I'm combining these two areas together, because many of the scientific concerns are shared between these two.
We have another question about the implementation of deleted process. What software is utilized to implement the deleted process?
We are trying to make our package, and because there's a paper we published here, and we are trying to making our package available, and because we understand that the computation for the deleted process implemented by the therapy could be intensive, and hope when the package is available and we can have a more convenient use in this tool, basically.
Okay, we have another one. So from the presentation itself, it seems like you are suggest model assist design have more options compared to model-based design. Is this the case?
I think it really depends. I think in the cell therapy, I like model-assisted design more. It's because we all understand the manufacturing for cell therapy is so complex, which meaning we often only having a very short range of the dose levels to be explored, like 3 and 4. It's very common number we see in the cell therapy trials. And also, as I just mentioned earlier, it's going to be even from the toxicity profile, we see some trials that the higher dose that's not necessarily having more DLT observed compared to the lower dose. And I think in that sense, the model-assisted design is going to have a more appropriate use here compared to model-based design, because first, you don't have a relatively sufficient number of the doses to well establish or validate your assumed dose DLT relationship. And also you don't have, with that said, I think the second point is the model-based design. The flexibility feature, we can still carry over from the model-assisted design, but just avoid all those pre-specifications.
Great. Next question is about slides page 28. I think this referred to TPI2 and UBI design. So can those methods be extended to handle more endpoint in like PK/PD and others?
Yeah, that's a very good question. And I forgot to mention that when we're talking about efficacy, we are talking about the programs or the pipelines that in general are expected to having the early efficacy observed. And with that said, directly using the efficacy endpoints, we along with safety is feasible for those studies. However, if you don't consider the response, for example, will be occurring for first month or second month or etc. And if you have already established some intermediate measures, like you mentioned, the PK/PD, and if you consider that those intermediate measures have a good threshold that you can conclude from the activity wise, it can also be utilized into the TPI2 and UBI, as long as it's in a binary fashion.
Another question is about, can we combine the measure you use for late onset toxicity for multiple endpoints?
We can, and they're actually, for the Bayesian ET that proposed by Takeda, they have a tied Bayesian ET as well. With that said, for our TPI2 and the UBI, we can also, in the likelihood estimates, we can put in the weight there and trying to consider the maturity of the patient's outcome. But we also need to remember that we are talking about a very small size of the patients in the early phase. So with that said, the more features may not necessarily going to provide you a more reliable profile. It may cause overfitting, or it may cause a hard justification about whatever the assumption you are putting there. So I think we need to think about in the phase 1 design trials in a sort of a more priority way. So we definitely have a laundry list, but we need to prioritize what is the most important questions, and then we can think about what design going to be better there.
All right, last but not least, I think let's one question from Kentaro Takeda, who actually have couple response answer online by different people. So look, but I think it's worthwhile to reiterate the question here. So the question from Kentaro is, have you already implemented F-Tox design in IO therapy trial? Does FDA have concern on the designs? In my understanding, the F-Tox trade-off design and the Boeing ET design has been implemented in the cell therapy program.
Yeah, that's a very good question. We haven't used the F-Tox into our internal trials, but I know MD Anderson have used the F-Tox design in one of their cell therapy trial. That's not a sponsored trial. So the process in terms of FDA review and the extent of FDA review may not be exactly the same as sponsor going to be having. But I think for the cell therapy in general, you need to think about if the F-Tox or whatever the model-based design is going to be working in a laser-focused way to capture your potential profile. Because in cell therapy, you may not see, sometimes you may not see toxicity at all, or you start to see toxicity in the early dose level. And the dose level, the number of those levels are very limited, right? So you need to justify why you consider such model-based design going to provide a reliable estimation. So with that said, when you justify, you probably want to use a simulation practice under different scenarios to justify that.
Great. Thanks, Rachel, for a very comprehensive presentation and answer to many questions. And also thank everyone for raising a lot of good questions in the chat box. With that, we would like to conclude today's presentation by Rachel. Christine, do you have any other additional to add for future webinars? Christine, we couldn't hear you.
Yeah, so just visit our website. We will be posting the slides and a link to the YouTube presentation recording of the event. Again, and we hope to see you at our future events. Thank you. Take care, everyone.
Thank you so much.
Thank you very much.
Yes, thank you, Rachel. Yeah. Thank you. It was great.
Thank you. Not very familiar with that. Okay, got it. I think I got it. Thank you, Kristen.
All right.